Hi everyone. Thank you for joining with us to this SNEV special series called Industry Academy Talks. In this talk, we participate Alexis Murillo from ECSP, Sao Paulo, Brazil, and I, Cristóbal Serra Troncoso from KU Leuven, Belgium, to present and moderate this talk. Called EVs from bench to bedside and back. And this opportunity, the talk was presented by Dr. Maroun Khoury. Dr. Maroun obtained his PhD at the University of Montpellier, France, and he then enrolled in a postdoctoral program at Massachusetts Institute of Technology, USA. And then he gets a position of the at MIT Singapore Alliance for Research and Technology, SMART, in Singapore. So in 2012, Dr. Khoury joined Cell4Cells as Chief Scientific Officer, establishing in parallel the Nano Regenerative Medicine Research Laboratory at Universidad de los Andes. Currently, Dr. Khoury also holds position as a Director of Impact, the Center of Excellence associated with Cell4Cells and Universidad de los Andes, and as a member of ESCT's Exclusive Strategy Advisor Board. So thank you so much, Maroun, for joining with us in this presentation.
Thank you, Cristobal, and the rest of the board of SNEV for the invitation. I'm looking forward to learn as much as to teach today, and I hope we can have a interactive talk. I have a lot of questions as well to ask during my talk, and then we can have the space to discuss them, and I want to know your opinion because you're part of that ecosystem of EVs, especially when we talk about translation going from academia to industry. Okay, I need to, all right. Let me put my slide on and then we can slide in directly to the talk. All right. Okay, can you see my slide? I believe so.
Yes.
Yes, yeah, we can see.
Perfect. So again, thanks for the invitation. The talk is how to get your technology, and in this case, I mean, we're in the SNEV, so how to take your extracellular vesicle technology from bench to bedside. Here I'm focusing too much on therapy, but then of course you all know that EVs as well are being used as biomarker, what they call liquid biopsy. And then our experience goes from, you know, the idea, we started working on this 7 years ago, roughly. And then our goal was to get them to the bedside. And once we were able to do that, which is not something easy to achieve, easy said, but not easy done, we figured out that sometimes you have to go one step back and then bring it back to the bench to fine-tune things that you think that are missing and are very valuable for the translation of that product. And that game of going from bench to bedside and back, it looks like the snail and the ladder. Sometimes you think you're ready, and then you're ready maybe for a proof of concept. Then for a phase 1 or phase 2, you have to go back and look more at the mechanism of action. And this is a bit of the interaction or the itinerary that we're following for EV to become a clinical reality. So I'm very interested in the talk today because this is a student network, and your role is very important in bringing any technology from idea to social impact. And there's been a lot of editorial, and I've picked up the one that might be the most impactful for you. It's Nature. We know how our favorite journal, where they wrote an editorial about PhD training is no longer fit for purpose. It needs to be reformed. It's obsolete, etc., etc. You might be agreeing with this editorial or against it, but what's the more important is how really you can use your PhD to get the impact more than just the publication, the dissertation to get your degree. And if you're a postdoc, just going through another round of new project publication, etc., etc., and to make it closer to the need, the industry need. And And the answer in that editorial is not answered. It's just give you an overview without giving real solution. And what you're doing at the SNEV is actually trying to modernize the PhD or even the student or the postdoc position in a way to integrate it more into the real life, the industry. Some countries, like in Australia, they created the iPhD, which is industry PhD, training students to be part of. 10 years ago, when I moved to Chile, and I had this dilemma of leaving academia, stepping into the industry as an important challenge, and I understood 10 years later that actually you can go to the industry, step back to the academia, do your bench work, go back to the industry, and it's not a end road. It's actually a feedback where you can go and come back. And there is no more dilemma of leaving industry, academia, go to the industry, and thinking that there is no way back. Industry and academia today are working more than ever together. And a absolute argument for this is, when you look at big pharma company, Pfizer, Lilly's, etc., GSK, they were viscerally, organically working on their new drug discovery inside. The 10 project, you will have 9 done internally. Today, if you look at the same company 20 years after, maybe only 20 to 30% of their project are done internally, and then the 70% are idea brought from the outside, outsourced from academia, from small startup, and now they're part of the ecosystem. And that is something very valuable in a way of reforming the old style PhD and postdoc. So plus you've been very creative in your logo. And then if you walk through Paris and you see that building, you will definitely think that someone of your member living there was painted exosome all over the facade of the building. It's actually the Japanese artist. I'm not sure if you're familiar with her, Yayoi Kusama. She's been very creative in painting those circles, and she made her style on this. And I believe she would be the queen of exosomes. Probably we'll have to reach her officially from the SNEV and tell her she's been painting exosome throughout her career without knowing it, and that gives even higher value to her art. And I'm mentioning this just because if you want to be disruptive, you want to really move from an idea to the market, you have to be disruptive. You have to be creative. You have to be creative from the logo, from the technology, to in a way to demarcate yourself from other. And this is a bit what I would like to convey to you today. So if we look at drug discovery strategy in 2023, we see an important shift from the chemical-based pharmacological traditional product to monoclonal back in the 90s, 2000, RNA-based drug with the COVID and the vaccines. And now we see that even traditional company, and I would say very traditional company, moving to EV-based drug. And I like to give example from the actual news. When you look at the investors, which are part of the ecosystem, and you should not be ashamed to at early stage going and see what investor like, not because you don't want to attain your science or your research with ideas from the entrepreneurship field. It's a market pull, and compared to the technological push that you are doing in your lab, so you are matching it, and this is the connection that is very valuable. I mean, the talk academia versus industry is all about that connection between your technological push, the stuff you're putting in your lab, and then the pull from the market where investors have an important role. And we can know that there is an important boom in merger and acquisition and licensing deal in exosome. The last 2 years, it's equal $1.5 billion, where major player like Lilly's, Takeda, and other big players are being interested in technology that you guys are doing in the lab. And then you can see as well companies who have failed their phase 3 clinical trial. This is November 2022. And then they said, okay, we still have 45 million cash on the hand. What should we do? Let's move to exosome. In that case, they were trying to find treatment for pancreatic cancer, was a really bad prognostic of 5% of survival after 6 years. So they deviated their strategic shift from a traditional drug two words, an exosome that can be delivered. In that case, just for interest, it's exosome secreted by red blood cells. And I should have started with that slide about my disclosure. I have an academia position. I'm a full professor at the University of Los Angeles. I'm part of the strategic board of director of the ISCT. It's another society for cell and gene therapy. 2 years ago, we were able to secure an important 10 years grant in Chile. This is really an important milestone for Chilean science, where we were awarded to establish a center of excellence for precision medicine and advanced cellular therapy that include exosome. This is why I'm here. So this is my academic part. And I do have as well industry position. I'm the chief scientific officer of a consortium. The first part were non-for-profit, and this one are for-profit. I'm as well the acting CEO of Cell4Cell, which is a startup company that came up from the same University of Los Angeles. And I do have as well, I'm authoring different and variable patent in the field of cell and gene therapy, including EVs. I'll be presenting some clinical data. So this is my speaker clinical data declaration, according to the Declaration of Helsinki. The preclinical, it's according to the Declaration of Singapore. So since it's a virtual meeting, I'm going to give you an overview, a virtual visit. This is our campus that lays on the foot of the Andes Cordillera, the Andes Mountains. This is our main building. Up there, this is our university hospital where we have our GMP. This is where our exosomes are being produced currently and being used for the clinical application. As I mentioned to you, from that ecosystem, the university, the TLO office, there was interest of a startup, building a startup for regenerative medicine. An idea is to reduce cost, making more accessible, and then skipping the geographical barrier. I know there are students from all over the world, from Brazil, Malaysia, Europe, and the US. So this is something as well you want to do for your own region. All right, so at Cell4Cell, we work on biomaterial, stem cells for different application. We start with heart failure. We published that in Circulation Research. It was a landmark paper that was published in media outreach in different countries. And then we decided that we have to focus on osteoarthritis, and then this is this strategic shift that we've done in this company. And today we stand on 10 clinical trials. Most of them are completed. 2 of them are ongoing. 2 of them are EV-based trials. One of them is on osteoarthritis that I will have the time to explain, to disclose. And we have another EV trial that will start very soon, in a couple of weeks as we speak. It's in the use of EV in dental pulp regeneration. So it's a very low, again, confined local application of these EVs. We have completed 3 phase 2 clinical trials. Overall, we have 800 patients treated with 0 adverse effect. Mostly they were treated by stem cells, and now, as I mentioned, shifting to EV in the different application that we've used. So why EVs? We have shown some interesting data, the use of the cells, but as you all know, stem cells have some limitation. Just to situate the battery of tools that we are developing for the treatment of osteoarthritis. I don't want to focus on osteoarthritis. I want to talk about the EV platform that we're developing, and osteoarthritis being a model that I would like to explain further. So in the field of osteoarthritis, it's a cartilage degeneration that can cause pain, lack of function, and motion, etc., etc. There are no cure drug. 20% of your people from 20 to 40 have osteoarthritis, and that can increase to 80% after 60 years old. It comes from aging. It's age-related disease, but as well, it's intense use of your joints, such as professional play, etc., etc. So orthobiologic are using what is the biology offers, such as bone demineralization or other issues. And then we have the live orthobiologics, which are the stem cell. You can use those stem cells alone or using a carrier. You have the micro orthobiologics, and in that case, we're using, for example, mitochondria that we isolate from the same stem cells and inject them into articular. This is still preclinical, but there are some clinical work in different applications using mitochondria. This is not the subject of today's talk, but it's a line of research that we're pursuing. I will be presenting it at the ISCT 2023 in May, end of May in Paris. And what's interesting for us is what we've been able to do at the nano-orthobiologics level, and by this I mean the use of exosome as an orthobiologic for treating osteoarthritis, right? So it's all started when we wanted to understand the mechanism of our first clinical trials when we used MSCs, when we understood that some of the mechanisms were probably the differentiation of these cells post-injection into chondrocyte, which is what we need because in osteoarthritis we have a degeneration of these cells. After many years of research, many work has evidence that MSC are able to differentiate to chondrocyte, but that does not explain their mechanism of action, which means it's a marginal effect. And we start looking for other effect. We look at the paracrine effect, and we start looking at secreted factor, which confer the immunomodulatory effect of these MSC. Later on, of course, we start working on EV packaging many molecules that confer this anti-inflammatory and hopefully protective effect. And finally, we as well start looking at the natural transfer of mitochondria, leaving the MSC and going to the damaged tissue. All these together might account partially or fully to the mechanism of action of OA. And then we thought beyond only looking for mechanisms of action, can we advance from cellular therapy to acellular or cell-free therapy? And by this, I mean the use of these secreted factors, including exosome, including mitochondria, as a therapeutic agent by themselves. And this is what we've been doing for the last years. So I want to get you back to the story where we were looking for what the best source of stem cells. And when you're working with EV, you might be as well working with stem cells if this is a part of your protocol. So when we started the project a couple of years ago, we were sourcing for the optimal stem cell source. Stem cell can be isolated from any tissue you can think about, even heart tissues. But then we wanted to look for a zero strategy, which means zero invasiveness. I don't want to have a operation to remove those cells from the patient, whether it's a punction or whether it's a full-blown surgery. So we wanted to have zero invasiveness. I want to have the youngest tissue possible. What is younger than newborn babies? And this is where we're going. And of course, the cost is zero because you're using actually medical waste. And this is what brought us to the use of post-birth tissue. This is C-section delivered babies. The tissue you're able to obtain is the decidual part, which is the maternal tissue, and then the fetal tissue. In our case, we've selected only male pregnancies, just because it's a tool for us by doing a fish assay on the Y chromosome to differentiate what is from the mom, what is from the fetal. And what we've done is a simple comparison study when we looked at the different tissues from the maternal side, from the fetal side, and from the fetal side, we've looked at different tissue, including placental, chorionic in that case, the amniotic membrane, the umbilical cord blood, the amniotic fluid. You have more than 5 tissues you can isolate. And then what you do is a neck-to-neck comparison towards the important potential that you're looking at for the application. So here, I'm not saying that the tissue we're selecting is the best source. I'm saying the tissue I'm selecting is the best source for the precise application that I'm looking for, which is the treatment of osteoarthritis. In that case, we look at the chondrogenesis effect, chondroprotective effect. We look at the immunomodulatory in different experimental settings. This has been published in 2015. And then when we selected the umbilical cord being, not the umbilical cord, but the umbilical cord, the physical cord, the Wharton jelly being the best source according to that pentathlon of potency tests that we've done, then we moved to test them into our mouse model for osteoarthritis. Actually, you induce the disease by injecting a collagenase. This is an enzyme that will erodate the chondrocyte within a few days. That will mimic what will happen in your patient knees, where you have chondrocyte and cartilage erosion. So this is where we start injecting at day 7 and day 14. In this case, we're injecting the cells. Again, I'm telling you the story where we started from the cells. This is then you can evaluate according to the clinical score, looking at histology. There are known scales that you can follow. We do as well mineralization. And in that case, the more mineralization you have, bluest the color is, the least is in reddish and then purple colors. If you're working on osteoarthritis, you wanna see less mineralization because you want to have more cartilage. If you're working on osteoporosis, then you will be looking for higher mineralization. So this is the difference between the different two models. So in that case, we want to have less proof of mineralization and more cartilage, more soft tissue. So long story short, we did different doses of cells. We injected 200,000, 50,000, and we'll be able to show an effect control compared to the sham control. Taking all these together, we've moved to a clinical grade production of these stem cells, and then moving directly to a phase clinical trial, phase 1A2 clinical trial, where we had 3 groups. We're comparing to the standard of care, which is at that time was the hyaluronic acid. It's been, the standard of care in OA have been shifting depending on the evidence and the meta-analysis done in the field. And we've done one single injection of these MSC. This is intra-articular injection, and we compare to double injection. And then this is a video that I'm gonna cut the noise. It's in Spanish, so I will subtitle it. So this is exactly what we've done. We've cultured those MSC from the umbilical cord, made sure that we were able to manufacture them at the clinical grade that is required for their use in patient. And then what we did is define a specific dose of cells, and then did the intra-articular injection. We're injecting a specific volume intra-articularly, and then we're following the patient for 6 and 12 months. So the idea behind this is that MSCs will go and then regenerate the cartilage. What we've been able to show compared to the control is a reduction in the pain, reduction in the rigidity of the articulation, and increase of the cell function. Of course, these are the data from the trial that was published in 2019, looking at 6 and 12 months, and where 2 injection showed a longer term of support. So this is following the track. We're going for a FDA IND approval as we speak. We have a 2-year follow-up beside that paper, and an IND filing in Chile after showing safety and efficacy. We're able to have a conditional commercialization. So this treatment is now available under specific condition in Chile while going for IND approval in FDA. We like these cells. That treatment has shown important effect on patient, but as you all know, cell therapy has important limitation when you compare it for other therapies, including EV-based therapies, where they have the advantages. I'm not gonna preach a convinced crowd here. You're all part of the SNEV, so you'll know that EVs are cool. But then as well, we selected 3 different parameters to compare cell therapy versus cell-free therapy, EV therapy, at the therapeutic level, at the commercial manufacturing, which is very important in terms of simplified logistics, ready-to-use product, and of course at the safety level, where these are inert particles that are not able to shift. The problem with MSC, if you put them in a high immuno inflammatory, they can shift from MSC1 to MSC2. That's a pro-inflammatory to anti-inflammatory phenotype. So they are influenced by the milieu. If MSC reach the tumor site, that as well can be switched to a tumor sustained cell. So something that exosome or EVs are not able to do, which is very positive. So from there on, we said we're going to follow the same track, doing preclinical, and this is our multidisciplinary team. Went back to the bench and then set up an EV preclinical team. I will go through the details about all the preclinical package that they were able to conduct. And then sometimes we think it's from the bench to the clinic, and it's not true. So it's from bench to a bioprocess lab, which is a lab that takes your lab books and then switch it, modify it, and transform it to a SOP, standard operating procedure, which make it a protocol that can be used in GMP or clinical grade. And then that SOP is trespassed to the clinical grade. So you're making a proof of concept of moving a protocol from your cell culture hood to the GMP space for clinical grade. So this sounds straightforward. I'm gonna tell you the truth, it is not. It takes you between 1 to 2 years of work to do the modification, the adjustment. Some products that you use in your lab are for research use only. For this means you're not able to use them in a GMP, and then you have to reuse all other product, and then to show that the final product you're manufacturing is not changed. This is really the hidden side of the interesting stuff that you do in the lab. Behind it, there are a lot of work that are required to move it to the GMP, and then of course to the clinic. And this is the team, mostly student, postdoc, working on our R&D team. And this is the 13 preclinical studies that these are the minimal assays that were required to be able to move that product from the bench to the bedside, looking at, of course, the typical characterization according to the ISAF society, but we have to take it slightly further. I'm going to mention some of them that are beyond the ISAF guidelines. You have to look for internalization in your target cells, the biodistribution, mechanism of action, stability, potency tests through cargo characterization, macrophage polarization, which is in our case one of the target cell that we are interested in, looking at the dose response in our OA animal model, immunogenicity, clinical grade production characterization, and finally potency test, quality control. So this is all the battery. And then before, these tests were done according to your PI interest. Probably he's more into the immunosuppression, so we'll emphasize on this, leaving behind other assays. And now, and this is part of industry academic talk now. The industry is a bit upset of unreproducible data that they've been diligent from the lab. So this means industry come to the lab, and this is what most of us want to see our technology being licensed. And then they look at your data, they think it's interesting, they think it's what they are, part of their investment scope. They give you an upfront payment, and then they take your technology with your help to their lab, and they try to reproduce it, and then bam, only 25% is reproducible. And this is I'm not talking about specific country, I'm talking about global, even from the best university in the US. And this means 75% of technology being transferred is not reproducible, or lacks technology, or lack the ability to become a product. And then therefore there are some lack of enthusiasm now of getting those licenses from the academia. And there is a lot of things that we can do to improve that. And I'm saying we found the solution. I'm just saying we actually understood the problem and acknowledged the problem, and now trying to find these solutions for your diligence. It's very important to understand the importance of reproducibility in case you want to license your product. So again, this is the protocol we've been using from the lab to the GMP. We've been looking at the different markers. So the summary of the preclinical trial, which are very important. The solution is to have as well a CRO can help you with your preclinical packages to make sure you're not missing any information, because any information means you're not going to be able to get your IND approval, and then you have to go back to the lab and then probably do 1 or 2 years of work, which might upset your investor, which might upset the person who is interested in licensing and will go for a quick and ready to go project. And then after 2 years, probably it's too late. So this is common, just to show you the data. We'll be able to show that they are being able to uptake by macrophages, by chondrocyte. This is by typical staining. You guys are familiar with these protocols. Again, using the OA mouse model with the collagenase-induced OA, we've been able to look at the induction of the disease, injection of exosome intra-articular. In that case, we've done 2 injections. So we have a scoring system, and this is what we're able to show, that EV's injection have significantly a lower OA clinical score. We'll look as well at the micro-CT for mineralization, looking at the color intensity, a different area, as shown here by these stars. And then showing significant differences in terms of efficacy. Again, to complete our preclinical package, we had to do the biodistribution assay, and here we're looking at the injection of labeled cells, and then when I compare cells to the EVs through intra-articular injection, looking at up to 72 hours. What we're showing you here is we do not see leakage, at least with that way of looking at the injection site and the fluorescent probes using local injection for the cells or for the particle. We do not see a leakage outside the injected. We don't see ipsilateral or contralateral, means other joints than the injected one, at least for 72 hours. So characterization RNA are part of the effect or not. It's a characterization step that is required to look for later on reproducibility, but as well you can, as you all know, use them as a way to understand the mechanism of action, the pathway that can be induced by these content cargo, looking at different pathways. I'm going to go quickly to this uncompleted bioinformatics studies where you can have a list of pathways that can be active in the cargo of these EVs. Again, this has to be validated, which is not at this stage. Then we looked at the polarization effect of those EVs when coupled with or were incubated with macrophages, and this is what we call a polarization shifting from a pro-inflammatory M1 to M2 under inflammatory condition, where you put your culture in LPS and an interferon, you add that to that, the EVs, and you look for this polarization, so a shift from M1 to M2, looking at specific market that can identify M1 or M2. And then this is where we want to use that beyond a lab test as a potency test, a test that can show that your final product has the effect, but as well can reflect or predict a clinical potential. Bringing you back to the most boring slide, but the most important slide, which is data reproducibility. If you're a PhD student, and this is a lot of work, months of work, and then it's actually not even a slide. It's probably supplementary data that can go through your report or your paper, but it's very valuable if you want to, license your product to the market. So what I'm showing you here is 6 different donors done at 3 different preparation, looking at concentration, looking at size mode. And what you can see here is if you have a good manufacturing protocol, you will reduce, you cannot eliminate, you can reduce variability between donor and as well between preparation and another one. Again, this is another proof of showing that markers are not changing, importantly, between one batch and another, between one donor and another. And what happens if it does change, then you will have to have selection criteria, and then you can eliminate this based on standards you have applied, percentage of markers, etc., etc. So you have to establish those release criteria internally and then abide by this. And then the cargo, again, if it doesn't explain the MOA, it could as well be part of the profiling you need to do to show reproducibility. Here we're looking at 3 donors, and then the top 10 microRNA presented in those cargo are the same in all the donors, which can reflect as well the reproducibility or the lack of variability of your product. Then we move to the GMP production lab. If you're not a bioprocess or manufacturing interest in, then this could be as well another boring slide, but in every step you have to do the QC. When you defrost and expand yourself, you have to do a batch of assays, mycoplasma, etc., etc. After EV production cell, you have to go to another set of sterility control assays, and then after purification, you have to do the full list of assays, which requires as well a lot of materials. And if you have scarce material, this is an extra work, and you have to expand even more, produce more EVs. This is part of the QC that we do for our sterility. What we do is endotoxin tests. Everybody is familiar with mycoplasma, but if you want to release it for clinical grade, you have to do as well endotoxin tests beside other sterility. This is the full batch of clinical control, quality control that you have to do for your final product. And then when you have it, you have your release criteria done that include, of course, the the flow cytometry, the sterility, the visual inspection. Then you are able to release that product for your clinical trial. So this is what I'm showing you. This is the first phase 1 trial with exosome-produced therapy. I should say EV. I'm talking to SNEV, but outside the very scientific technical field, exosome is still a word that is more familiar to our investors and VCs. So this is a 10 open label, 10 patient, 12 months follow-up was a defined single dose injection. This is for safety mainly, but as well, even if the first endpoint is safety, our scientific curiosity want to go further, and they want to know if we have as well efficacy. So we have our inclusion criteria, and this is the eligibility criteria to be part of our clinical trial. No infection, etc., etc. So as a proof of concept, we were very anxious of launching a phase 1 trial before having a proof of concept case study in one patient. And so we started a one case study. It's a female patient, 66 years old, with symptomatic knee OA. It has a grade 2, was non-responsive to traditional treatment, corticosteroid or hyaluronic acid. So she was a perfect candidate to start this trial. And then again, this is the score. This is for pain. So the higher the score is, the more pain you have. We're showing stiffness. The highest score is the more pain, and then the function, the highest is the worst. So after 6 months post injection, we have a complete reduction. And then 1 year after, you have a small bump, but it doesn't reach the initial, which means this is injury, which means that it's EVs might have a therapeutic function, and then probably we will have to consider the timeline between one injection and the other. Of course, that single patient is not able to answer that question. This is definitely not a final proof of the use of EVs in OA. Is just a emotional and then motivational argument for us to continue pursuing our investigation, our R&D line. This was presented at the ISAV in Lyon last year, and at an ISCT in San Francisco. And then this is a kind of where we stand today. We need to validate our potency. This is the back to the bench. After continuing the phase 1 study in parallel, we're looking for validating our potency tests, linking them to the therapeutical effects seen in the clinic or not seen in the clinic. Today, there are no commercial scale of EV production, even they tell you they are, at least for following our protocol, which means lab-produced EVs. We have to understand more the product shelf life when you thaw them, how much time you can leave them on at room temperature before injection, how much you can leave them in your tank, etc., etc. Of course, there is an interest in modifying these EVs. Can we combine them with current drug? There are FDA approved, there are used in the clinic to potentiate the drug. We're talking about a synergy. We don't want only 2 EV to be better than traditional. We might work together in a synergistic way, improving their effect, but as well reducing their side effect. Some drugs have important side effects, and this is why they are not used in the clinic. And of course, today there are the field is going to the parental cell that are producing the EV can be immortalized, and there are pros and cons to this, including regulatory aspect that you have to look at. How much time you will spend if you have immortalized cells versus non-immortalized cells in an IND application for getting approval. So in a signature series that was done last year by the ISCT, where we were able to participate, this was published only last month by the EV committee, led by Bern Gibel and then Sai-Kyung Lim from Singapore, where the conclusion, I'm showing you the conclusion of the different talks of people who, like us, were able to bring their technology to clinic or close to the clinic. And then the conclusion is, while there are interesting advances, there are still a lot of questions to be answered. And they're similar to my previous slide, where biodistribution is important, looking at the pharmacodynamics thoroughly, looking at the cellular fate, after the update, the root of EV administration. These are from the conclusion that all these members from academia, but as well, we have a lot of people, I would say 70-30 from as well industry, showing and explaining giving their conclusion, including the engineering of EVs and whether that changed as well the uptake and the fate of these EVs. And I want to finish, not with the ISCT or ISAV or cytotherapy or any other reference. I'm going to use a kind of surprising reference here. It's from the White House. This is September 2022. It's an executive order. It's what we call EO, signed by the President of the United States, Joe Biden, on advancing biotechnology and biomanufacturing for a sustainable, safe, and secure American bioeconomy. So this is a 65-page document. If you are interested in, I can send it to you. It was sent to me by a colleague in the U.S. And then this is very interesting because it has 5 chapters, including food, including environmental cues, but as well, this is why it's interesting. This is an article published as well this year about why we should answer to the call of this that executive order. So the bold goals include one chapter on biotechnology, on cell therapy. It doesn't mention extracellular vesicle in his summary, but it does subline it in different part. And this is the conclusion from that executive order, where the summary is that we have to improve biotechnology and biomanufacturing. That boring slides that we always want to keep in supplementary data that is not interesting for our high impact papers or our scientific curiosity. But this is what it's aligned in that document, that in 20 years, increase the manufacturing scale of cell-based therapy, including EVs, to expand access, decrease health inequities, and decrease the manufacturing cost of cell-based therapy tenfold. They put a landmark. We want to decrease it for tenfold. Just because you have to ask yourself, those amazing advanced therapy that we are very happy to see done by many groups, including labs probably that you are currently working in throughout the globe. The question is, who can access these therapies? And then how we are able to decrease health inequities. I work from a region in Latin America, which is Chile. Chile is escaping. It's one of the most important economy in South America. But then we have country that has low-income countries, and then we know that they will not be able to access these treatments. Even in Chile, we have problem accessing advanced therapies. So this is where our center, Impact, is a non-profit organization. So I'm not promoting something for profit. It's a non-profit organization. It's a center for interventional medicine for precision and advanced cellular therapy. This is why we call it Impact. And our main mission is to make those inaccessible, hard to get, complicated to simplified, accessible treatment for our society. And I'm sure that can comply to as well your proper need in all this. So this is a team, and this is my final slide because we want to leave some room for discussion. I haven't been looking at the hours, and you guys have been very quiet, reminding me the time. So I think we're just on time. So this is the team behind all the work and the effort I'm showing you here. And we're part of this ecosystem between the university, the Center of Excellence, and the startup and the consortium coming up from that hub. So thank you very much for your attention, and ready to take your question and ask some as well for you. Thank you.
Thanks so much for your presentation. Very nice talk. Yeah, first question about the participants.
Who's still Alexis? You have a question?
Well, now we have a space for our participants. You can send your comments, doubts, suggestions, curiosities, etc. for Dr. Khoury. Only reminder, you can write your comment in our chat box, and I will read it later. or just raise your hand to take the microphone. Okay, we have one question here from Chiyong. Just let me tell you. Thank you for your interesting presentation, Dr. Khoury. Here are a few questions from me. One, there are few studies showing that US MSCs have lower chondrogenesis ability compared to other stem cells. May I have your point of view? And second, may I know how do you determine the optimal dose of small extracellular vesicles in your animal studies and clinical trial?
Yeah, it's a very good question and very interesting, especially the second one. We've been trying to answer that for the past 3-4 years. So I'll start with that one. In our preclinical model, the volume, the answer of the optimal dose was very simple for us. Of course, we can do all these stoichiometric calculation, number of EVs per cell, per volume of cartilage, per number of circulating cells, immune cells in that cartilage, and come up with an algorithm, AI based or non-AI based. We followed a simple physiological cue, and this is the maximum volume. You know how much you can inject in a mouse knee joint space? Up to 5 microliters. Actually, it's between 3 to 4. So we said how much maximum we can pack in that 3-4 microliters of exosome through the current technology. This is going to be our optimal. And then we can start decreasing it. And this is how we decide our optimal based on a physiological cue. And then we start to expand it to extrapolated to human or patient size knee. And there are definitely more, experiment to be done to looking for the optimal dose. Right now, for the limitation was the volume of the joint space in our animal models. And definitely after the phase 1, we're going to have that question being asked by our regulatory board, and then we will have to do a dose de-escalation and escalation. First question is I'm going to say eventual lower and higher it's going to be depending on your experimental setting. If you're taking an experiment done in lab A, compare it with an experiment done in lab B, not neck to neck, not within the same condition, then it's not a good comparison. What we've done in our case, we've taken bone marrow, we've taken fetal tissues, we've taken maternal tissues, total of 5 different tissues, and we compare them neck to neck in a precise experimental setting. And in that precise experimental setting, we've shown that umbilical cord MSC have higher, not only chondrogenesis. Chondrogenesis is interesting, but as well chondroprotective, something that I haven't shown here. So the ability of protect degeneration. If you want to look at other sources like IPS, MSCs, for example, if this is what you're hinting at, then you have to do it within the same experimental setting. And then I'm going to tell you something. It's not going to be accurate. You know why? Because I can show you a comparison between umbilical cord MSCs and umbilical cord MSCs, but they're from different donors, and they can have a different outcome. So what you have to do is you have to determine how to erase that donor to donor variability by putting inclusion criteria, by putting QCs, and by putting appropriate potency tests to avoid having that comparison. iPSC MSC donor A could be better than UC MSC donor B, but vice versa as well. So this is a very tricky, question to say it's higher or it's lower. Otherwise, we'll be comparing apples and pear, but it's definitely a good question to ask.
Okay, thank you, Dr. Khoury. Thank you, Chu. I don't know if someone wants to comment to add something about
Yeah,
this.
I want to ask a question. Actually, this topic is very interesting for me. I'm also, my research area is the therapeutic use of EVs. And I applaud you for covering this whole topic in a short time. I know it took you months or maybe years to come up with all of this. Covering from the scientific aspect, then to the industry, then to the inequity and how this could be translated, as you said, from bench to bed. And so my question is very basic from the beginning. How to choose a functional MSC? From research, we know that not all of MSCs are functional. So do we have to try one by one, or because there are no markers to check for the functionality of MSCs or EVs after that. So how did you solve this problem?
Oh, we did not solve it. We understood that one test is not going to help us determine precisely the functional effects. So what we're working is a set of experiments, and it's burdensome when you want to do a production and you want to release it. Sometimes you have only weeks to release it because you have already enrolled your patient. Ideally, I would like to have a single test, but in the real world today, as we speak, we have to have a battery of assays put together, and then all together they can answer whether these are functional or not. We're working on a potency test, and I put question mark because we believe that this could be a valuable potency test. But if you look at the industry for the past 10 years, a potency test has been a headache for stem cells. And then we want to learn from our ancestors' mistake, ancestor of EVs being the stem cells. And we are trying to prevent what happened to stem cell. I'm going to tell you exactly what happened. This is a very interesting lesson to learn from something that happened only a few months ago. We have a Australian-based company who have advanced to a phase 3 clinical trials. Phase 3 clinical trials, we're talking about millions of dollars of spending, right? It's 20 to 60, even more. They were working on stem cells for GVHD, graft versus host disease, for their pediatric use. They reached their primary endpoint, right? Survival. However, FDA didn't authorize their phase 3 clinical trial. Why? Because they didn't have a proper test. That is a big blow in the industry. So you do a phase 3, you're able to get the investment you need. You execute it perfectly. You get the outcome, the primary outcome, and you don't get clearance. And this is mostly because of not being able to have a well-characterized product that is not variable from one batch to another. This is how important it is. So now what's
and it's difficult, right? Yeah, sorry. But it's difficult, I mean, because of the heterogeneity of the humans, right? So
yes, it is. I'm talking about the cells, and the cells are heterogeneous, and the heterogeneous cells give even more heterogeneous EV product. So you have to live with nature. We don't have to, of course, we can say, let's get the purest EV. Even though that one, we can show you that it's not that pure because you will still have heterogeneity in the cargo and not in the surface markers. So maybe we have to live with that. And probably heterogeneity is not our biggest enemy. Our biggest enemy is variability. If you have the same heterogeneity, and then let's say seven, I like your logos, so I'm going to use it as an example. So if you have 70% of red dots, 20% of blue dots, 5% of green dots, and 5% of everything else, if you want to maintain this towards your production, and if you're able to show that you have that same profile throughout 20, 40 production, and then you're getting the same outcome, then, well, let's live with the heterogeneity, and we don't have to get the purest. The problem is when you have variability, and now your product is not 70% green dot, it's 70% yellow dots. And this is where it can give you, a difference in clinical outcome. So variability is our enemy, not heterogeneity. If you're able to produce it in the same way and consistently, then it shouldn't be a problem. And then when you change the production, if you move from chromatography to tangential flow, it's perfect. You're solving a scaling problem, but you want to show that the end product is similar. And that is what we call homologation study. It's easy said, it looks like fun, homologation, like, oh, product A's look like product B, but it's not, because you have to validate it clinically. So you have to go back to the clinic and then redo some clinical phase to show that this is the same. So what is my conclusion here is spend time in your bioprocess, biomanufacturing, because downstream, any change is gonna cost you money and time, all right?
yeah, I agree with you. Thanks a lot.
Thank you. We have more comments and questions here in our chat box. From Junju, thank you so much for your nice presentation. I would like to ask which kind of extra vesicle quantification methods do you think is reliable in clinical setting?
Right. I mean, the question is what are the available technology today and how that can be improved? The good thing is quantification is a downstream media. So it's not part of the manufacturing itself. It's only a part of the quality control. So any new technology can be added without having to redo a lot of clinical trials. We have to do in homologation studies in vitro or in vivo in the lab. So that's a good part. So right now we're using the MTA, and then we have acquired a new equipment that has a specific hardware. It's a flow cytometry that is able to detect up to 17 nanometers. And we think that with that flow cytometry based characterization and quantification and with MTA altogether, we can get a better insight on the heterogeneity of our product, because you can label them, but as well about the quantification. This is something that we are currently working on. It will not be part of our phase 1 trial, probably it will be part of our phase 1B. So technology is the answer.
Okay, thank you. We have more questions here from Nor. This is a friend from the group, and just from Malaysia.
Yes.
Thank you for sharing with us this very interesting talk. My questions are: in these studies entering clinical trials, we also can see some promising evidence supporting further growth of this EVs-related research. On your point of view, how far can this go? Can EVs replace current therapeutic approaches and give benefits in medical and dental work?
Okay, so this is interesting. Plus Noor Hayati is putting medical and dental. So this is very interesting, because there are as well a lot of interest in using those EV in dental application, whether it's endodontics or others. So I hate to speculate. Now because I'm part of a pipeline that is putting EVs in the clinic, I wanted to solve all our unmet clinical need, and it's not going to work like this. Again, I use my experience. You don't want to do the mistake twice. So I use our experience and our mistake done in the use of MSCs in the cell therapy pipeline to try to circumvent some of them in the EV. So we are well more prepared. So we think we can go further than what we've done with cell therapy. That's number one. But as well, we can tweak the technology, looking at why MSC did not reach important milestone. Today, MSCs have only few indication, whether it's in Europe or in the US or in the part of the rest of the world. So when we started working with MSC, we thought it's going to be the solution for all our problem: degenerative medicine, neurological disorder, inflammatory diseases, and even COVID. And then when we realized that they have only one thing they can do well, there are good immunomodulatories. They can control the inflammatory in some cases, but they haven't shown evidence of tissue regeneration per se. They are working on. So the limitation is because they were using native MSCs, and now what we're doing, or most, including our group and others, are trying to modify these MSCs. And there are two ways of modifying them: doing preconditioning, putting them in specific condition prior to their use. It could be environmental, low oxygen. It could be drug-based. So you put some FDA approved drug on them, you prime them, you prepare them, you license them, depending on what modification you are doing, or you can even do genetic modification. And then those products are showing better result or better perspective result. Now, going back to EVs, we can learn from that experience and say native EVs might have the same limitation, and we can have a proof of concept with native EVs, but then the way to go is engineered EVs, where you can have all these technologies, whether you're changing their surface marker or you're changing their cargo, their content, you're loading them with drugs, you're doing any modification for their specific homing. I think this is the product that can go further in the development pipeline, of course, with many limitations that we have to tune, right? But I think the field is not only on number of paper published, but as well number of acquisition. All of them were related acquisition. I'm talking about merger and acquisition, licensing from big pharma company. Most of them, if not all, were made from modified versus native EVs, and this is where the field is going, and this is where I think we'll see more effect in terms of the clinical application.
Okay, thank you, Noor and Dr. Khoury. We have more comment here from Chi Jong. Thanks so much for your answer, Dr. Khoury. This leads to my third question. Regarding the batch-to-batch variation, do you think immortalized cells will be the best choice for cell or EV-related products? Due to the heterogeneity of cell source, if I have no wrong, FDA will not approve if the consistency and sustainability of the product cannot be confirmed.
Yes. So I left that as an open question in my conclusion slide. I know many groups are moving toward immortalization of their MSCs or other source of cells, and it does have a science argument behind it. You will have single cells, they will not change through passages because MSCs, yes, they do age, and in our case we only use them from P6 to P10, so that gives you a short window versus immortalization cell. They can use them for longer, so you don't have to change donors. In our case, one donor is only solvent dose therapy, so it's still limited. You have to think that solvent is nothing because you will spend most of it in doing phase 1s to 3 clinical trials. So immortalization answer part of that question. The second part is what would be the effect of that immortalization on EVs, right? A short term, a long term, and this is something that we need to answer. And what is the regulatory agencies in your countries position to the immortalization? They might like it, but even if they like it, they're gonna ask you for more data, more safety data. They want to make sure that there is no byproduct, undesired product from the immortalization going through your EVs. And then that is not something so easy to show. You have to do longer term studies, and probably that will extend your preclinical package from a couple of years to many years. So you have to weigh in balance between the pro and cons, and this is exactly the open question I've put in. For us, we're not using immortalization. This could be something down the line where we'll have an engineered product that will have already complication in getting and showing safety and approval. And then if you add to it immortalization, then it all goes in one hand of doing modification and immortalization. So you're already in the non-native section. So the complication is kind of absorbed between the engineering and the immortalization at the same time. For native, if you're doing native, I don't think it's strategically interesting to do immortalization. If you're doing engineering, then probably it goes within the line of having your life complicated already, and then that doesn't add too much complication because you already will have to do a larger set of experiments. So this is my take on strategical take on using immortalized cells.
Okay, thank you, Doctor. I would like to amend this question with other ones. I am wondering about ancestry. As your EVs are provided by specific donors, not necessarily related with patients, do you believe that ancestry-related factors can alter results in some group of patients, or on the other hand, do you believe that the specific selection of some cell markers for producing extracellular vesicles could be a benefit to use them?
So we're just to make sure I've understood well the question, because if I didn't, means I already have another question to ask. So we're talking about matching donor versus non-matching donor. We're talking about allogeneic. This is the question: is how good it is to have non-matching donors? How important it is to have them match? If that is the question, or did I jump somewhere?
If we can have some compatibility problems between donor and patient.
Okay. No, Alexa, I did understand then well the question. That again, cell therapy or cellular therapy came from cell therapy, and then again, I always when I use those comparisons, because there are a lot of things to learn from the use of cells versus now use of their byproduct exosomes, EVs. So when you use the cells, and there are some preclinical work comparing histocompatible MSCs versus non-compatible MSCs. At early time points, if you're looking, for example, I don't want to add more models here. If you're looking at OA, osteoarthritis, when you inject MSCs from matching donors or non-matching donors, you will get the same effect in a short time period, right? 6 months. You're able to see the cells, whether they are from same donor or non-matching donor, for 6 months. And then when you're looking for 1 year or longer time points, you will see only persistence of matching MSCs. So the answer is, when you're using cell therapy, you might have an advantage when you're using matching versus non-matching at the long time points. But is that important? It's going to depend whether the major effect is produced at the short or at the longer time points. And this is how you will be able to understand whether it's an advantage or not an advantage using non-matching donor. For EVs, I think we already have the answer. The EVs have a direct effect. You inject them, they are internalized by the cells, and they promote that effect. So you don't want them to be hanging around. So therefore, I think matching is not an issue for EVs. For MSCs, is depending on the timeline of your experiment and then the effect that you are looking at. Now, cost-wise, finding matching EVs is going to be an important cost barrier. And even if you win a bit in effect, so let's say the matching will get you a year effect versus 6 months effect, it's going to be 10 times more expensive. And the question is, what is more efficient, to have 2 injections that has 10-fold cheaper cost than a single injection that can last longer? So there are variables beyond only the science, and this is very important, because when you are working in the lab, sometimes you're focusing on the scientific outcome, which is great, but as well, you have to think about the market pool. You have to think about the downstream technology. And this is not because it's an executive order that says technology should be 10 times cheaper, which now it's a good argument for me, but it's for making your technology impactful to the society, making it reach. A lot of technologies are amazing, whether it's in cancer, whether not application. The problem is not being able to make it feasible. And listen, look what is happening now. Advanced therapy products, if you look at the top 5 more expensive, you have gene therapy based treatment or gene and cell based treatment in specific indication that costs $2 million per patient. Maybe with the US health system, you can kind of get reimbursement, you can get away with, but then who can afford a $2 million treatment in other countries? And I'm including Chile, Malaysia, the rest of the world. So having those inaccessible technologies is as if they did not exist. And I have 3 examples. When you have $1.5 million technologies that got market authorization, it got the FDA approval, it went into the market, and I have 3 examples, and it crashed after 3 years. Not because it didn't work, just because the business model was not correct. The reimbursement model was not ready. So these technologies did not survive the market. So this is a point that you have to take in consideration when you want to develop something. It doesn't mean you should not ask that question. You will have to ask that question, but you will have to understand that question is limited to a scientific curiosity rather than to a strategic benefit and fitting. So this is the overview of that interesting industry / academia. And the good news here is 10 years ago, when I was giving talks about cost of technologies, cost of your therapy, people will come to me, hey, this is not my problem. I'm a scientist here and I want to work on my technology. Now the mindset, and you guys, and I call it the new generation of EV specialists and advanced therapy specialists, you already start integrating this from upstream, from the conception of your experimental design, which we call QBD, quality by design. And this is very positive. I will see that your technology will reach at least licensing point at a higher level because you're integrating those strategic cost question at early stage. And I believe that this is part of the effort of bringing advanced therapy, whether it's EVs or non-EVs, to the market in a more efficient way. And this is my forecast for the coming 10 years. We're gonna see those cues being integrated in your experimental design models, and you're gonna be more successful.
Okay, I got it. So we can think in cost benefit and evaluate the results of our technology. Thank you so much, Dr. Khoury. I don't know if you have more questions here. Let me check the chat box. Okay. Oh,
Thank
no
you.
more questions. Okay.
Can I have one? Can I have one last? Sorry, Alexa, can I interrupt you? Because for me, talking to SNEV is about, you know, giving information, but as well learning from you guys. So I have one question. That's maybe we can do it with hands up, and I'm not sure if we can use the poll. It's complicated. Let's forget technology and then have a quick vote. I have a poll question. If you have $1 billion, of course, if you have $1 billion and you're a student, you will pay your loans, you will pay your house, you'll buy a car. But then let's say if you're sitting on a venture capital board as a scientific advisor, this is, by the way, a very good job, well-paid job. So you have to think about it as well if you want to move to the industry. So if you're sitting at a board and you have to make a $1 billion decision where to put the money, will you, in EV, of course, and this is your expertise, right? Would you invest in EVs as biomarkers, or you will invest at EVs as therapies, right? So let's make it as a poll. You'll have your reaction. So who would invest $1 billion on biomarkers, right? And then we can subtract. We have 11 participants. So who will invest in biomarkers versus therapies? All right. Do I see hands? None of you guys? All right. All right. Who will invest in EVs as therapies? All right. So we have a bias here. So 1, 2, 3, 4, 5. So we have 5 already. 60% is theranostic related option. Of course it is. So well, okay, we have 50, and then the remaining ones, what would they invest? Probably 50-50, right? They will invest $500 million in biotherapies in biomarkers, sorry, and 500 in therapies. And I think it's fair enough. I mean, I think you guys had got it right. So they are interested, and investors are actually doing both. Biomarkers is already reaching the market, so the market is crowded because, you know, if you're a new investors and you want to see what's happening, and you see there are 50+ companies already using liquid biopsy as a technology, and then you want to invest today, which means you're probably going to be in the market 10 years. So you came slightly late, but still there is room for investments, yes. And then if you're coming to the therapy, it's, you know, there are more space, and then there are fewer therapies in the market today. There are at least real therapies. There are way fewer, so probably the investment is worth the effort. All right, thank you for
diagnostics is the way, right?
Yeah.
So we need diagnosis and treatment. Well, thank you so much.
Right.
Let me share here briefly. Before ending the transmission, on behalf of the SNEV executive core members, I will invite you to register and send your abstract to our virtual conference, which will happen on July 13th and 14th. Please look at this our social media for more information, and also you can just scan this QR code for registering and or to meet your abstract, or also to be part of the SNEV community. So that's it. I don't know if Cristobal, Memona, someone want to add something before closing this transmission.
Thank you so much for the interesting talk, and it covers everything and it gives us an idea of what's coming ahead our way, especially at the therapy area. Yeah, so thank you so much for your time and for all the inputs there.
Thank you so much for your time, and also for show the work in Chile, you know, because I think this is the first talk of South America in SNEV. So yeah, it's we are interested to show the EV research industry and everything around the world. So that is so nice.
Thank you for the invitation, and I'll be following the SNEV activities. I will push my students, volunteer, but I will push them to
Yes,
send.
please.
Force
And
volunteering.
if they are like interested to be featured in our newsletter as well, they're welcome to send our email, and we are going to introduce them to the community of EV students.
I think it's a win-win thing.
Yes.
I will do
Okay.
Absolutely. Well,
Care.
again, thank you for
Keep doing
joining
it.
us. We're very promising results, and good luck with your next experiments.
Thank you.
Thanks
Goodbye.
a lot. Okay, bye-bye.
Bye.
Thank
See
you.
Have a
Bye,
good
everyone.
day.